Okay, I'm going to start. It's not, hopefully it's not, still trying to. Okay, so today I'm going to discuss about chapter 15, functional modulation of receptor proteins on cellular interface with optogenetic system. And one good news is that my videos have like the English lines, so that I think that might quite helpful for audience to understand my English. Sometimes it's really hard for me to make the correct pronunciation of those words. So it's very helpful. Actually, before I'm thinking about I do that by myself, like adding those lines all by myself, and that's a great work. Well, I'm just thinking that this year I would try this all the book, go through all the chapters, and next year maybe I would like to update and improve my lecture. But it's so nice that the YouTube automatically give me those English lines. It's very nice. So today, we're going to talking about membrane signals and to go through some methods that using light to control the chemical transduction within the cell or mechanical transduction within the cell. So in the beginning, so for like complicated creatures like human beings, some mice, and some else that we compose of several different cells. And all of these different cells forms tissue, tissue forms organs, all the organs need to function together and they need to coordinate it. So just raise an example, like how the eye developed. In the beginning, it just from one piece of this tissue, then it forms like a crosstalk with the other tissues, and then it will affect like the upper side, some sides like developed into the retina and IPE cells, and some developed the choroid and other like blood vessels tissues to support the function of the eye, and some like be engulfed into the eye and form the lens, and this tissue is coming from ectoderm. I'm thinking yes, it's coming from ectoderm. So it's a quite so the development or the function of biological activities is quite coordinate and it's a complex function. And how this is so usually it's important that those neighbor cells talk to each other and how they function on each other, and it determined the final fate of those cells. So during reading this book, I'm thinking about what's the similarity of the light and the ligand. They all have like the Li that in at the beginning of the words. Some proteins like rhodopsin using the light photons to be its signal, and some use other molecular as their ligand, like a glutamate as their ligand to activate the function of the whole big proteins. So for generally for the chemical signal transductions using the G protein coupled receptor, a receptor tyrosine kinase, and ligand-gated ion channels, and these are all involved in chemical signal transduction. Yeah, and so we need to still go through to take a look at. And again, of course, okay, so in this chapter, the authors will discuss about mechanical the signal transduction and crosstalk between the cells. And so in the beginning, I'm thinking about for the mechanical like signaling, I'm thinking about the hair cell of the inner ear. So for the hair cell, so this is a cross section that shows the anatomy structure of the hair cell. Actually, so for the ear, that so this and is an image of the hair cell. We can see that lots of series. There's a very thin lines, and that's a tip link between these two cilia. And after there's a signal, sound signal, and it will change the plasma membrane and forms like the positions change, and this change was being linked by these tip links, and it will open to the mechanical motors. And this is kind of like these two cell compartments that are linked to each other, and the relative the positions exchange or change will open the channels. But there's other mechanical receptors that are involved in the signal transduction between the cells, like cadherin and intergrin. So for the cadherin, it's really involved like with to form the shape of the cell and to link different cells together, and it will also function together. So this cadherin is a transmembrane protein that forms an adhesive complex at the cell-cell interface. So within the cell, the cadherin were linked to the actin myosin complex, and it forms the tension of the intracellular parts of the cell membrane. So without the cadherin, the whole cell's morphology will be disrupted. In presence of calcium ions, extracellular domain cadherin dispersed on different cells form rigid contact between cells. It's exactly like this. When there's a calcium and cadherin or bind each other together, and all this is like some hooks and will hook together between these two cells, and how the cadherin can like being linked to the plasma membrane, the plan it's binding with the actin filament, and these connections were very tightly hold the plasma membrane and the whole cell with the cadherin, and the cadherin from neighbor cells forms the binding interactions or forms how the neighbor cells are linked together and forms biochemistry response that are coordinated in adjacent cells. So this is other. So within the cells, the cadherin were formed like the interconnection with the actomycin complex, and this within the ATPs, it will change the tension of the cells within the cells and the cells plasma. And this is here at the tight junction. Integrin expressed on the plasma membrane form rigid contact with the extracellular matrix, ECM, while the extracellular domain. Those integrins were forms integration with the extracellular matrix in the tissue, and they cause the cell's membrane to be like a seat on the tissue or within the extracellular matrix. And sometimes a disformed matrix will also cause were also very active, like binding or dissociate of the integrin and with the extracellular matrix. Using these mechanical receptors, cell directly responds to the mechanical cue in the surrounding environment by mechanical transduction. So in the latter of this chapter, the authors were talking about a lot of optogenetic control of those chemical interactions and the signals involving like GPCRs and receptor tyrosine kinase. And finally, the authors will come back to the cadherin and integrins and talking about how about using light to control the cell's behavior or interaction with other cells or with the extracellular matrix. So the authors will list all those candidates in here, like some from rhodopsin from the cattle, from mouse, human, from zebrafish. And remember, before we talking about the pyropsin, it's binding to all trans retina. And OPN4, it binds binding with a retina, but it will form and automatically, like it will not dissociate with the ligand or dissociate with the chromophore. It will automatically change within the dark, and they change to the active form of the 11-cis retina. So first, let's talk about how using the light to control the function of G proteins. First, let's talking about the melanopsin. Melanopsin, the chromophore melanopsin remain attached after photoreceptor and show bistable photocycle. Melanopsin is more resistant than rhodopsin, usually photoverse at different wavelengths. For example, the longer wavelength isoform derived from mouse and human function as a traceable pigment, which activate by a blue light and activate by a red light. And the pyroxenes, and all these can be activate different G proteins like GT, GI, GS, GQ, and the peroxines. And this image is from chapter 8. For the vivid is a circadian clock protein vivid from the foraminiferous vivid. It's also from homodimer. So it's activated by light and can form a dimers. And the CLY2. CLY2 is very important, very interesting within the activation of the blue light. Either it can form a dimerized with another CLY2 molecule, or it can form a heterodimer with a CIB1. So this reverse at the C-terminal surface, it has a PHR domain, and within the PHR domain is a binding the light response the chromosome. And it can also associate with helix loop helix protein, CIBI1. So later we're talking about how to induce mutations into the CRY2, then to determine and its function to be a form of homodimer, a form of heterodimer with the CIB1. So here it shows many chromosomes that are used by those optogenetic tools. And see that the phycochrombians, BD, antabrofins, and the PhoCl chromosomes. So here is some candidates that been selected by these authors as for light dependent interaction module. It means that within the light, with the light stimulation, for this is for a candidate for hoping that those protein forms interaction. And also there's a light dependent dissociation modules. It will show up here, and we're focused on this and also this. And with all that, with the light stimulation, it will cause dissociation of the proteins. And for the UVR8, its chromophore is this tryptophans, and shown up here. It is a chromophore. The dimer UVR8, which interact with arginine glutamate as paragon residue at the dimer interface, absorbing UVB when you take the absorbing UVB light by intrinsic tryptophan residues. Namely tryptophan 285, tryptophan 233. Also, another chromophore is the B12, as cobalamin. Okay, now let's move on to see the interaction with the ASLOV2 interacts with the JDK. So these systems, upon the illumination, it will forms protein interaction. It forms dissociation and separate to each other from LV2 to the JDK systems. And this, with light dissociations, can using light to control, like for example, the T cells receptor that dissociate with a specific plane or laminar or material specific, and it causes inactive or dissociate. So this PhoCl is another systems, and it has a protein. It's like a dissociation and cut of the chemical bond within the structures. So at the beginning, it shows a green light, and there's a chemical bond here. So after shedding with 400 nanometer light, and this bond is being separated, and this is being bracked. So this two part is being separated. We can also see here these two parts separated, and this bond is missing. So it shows a green, magenta color of the light, and it will later it will dissociate from the barrel of this protein. So this might be like a ubiquitous dependent protein degradation system, or maybe others, but it will dissociate with these structures after shedding the light. So there's a chemical bond or it's been bracked. So for these functions, so with shedding the light, you can control that some part with this nuclear export sequence or nuclear located sequence, and those two will be separated. So these two separated will cause the cell cause like the proteins that's located within extremely only within the cells, or after the light shedding the light, and it will coming out into the cytosols. So before it can be trapped by these protein structures, but after shedding the light, it can induce a translocation of the proteins, either because of there's a chemical bonds is break, and that's the PhoCl system. So now let's move on to go back to see the G protein coupled receptor. For G protein coupled receptor with a light stimulation or ligand, and it forms a different structure. So for opto XRs, and actually we're talking about this before, it's using like the extracellular part, we changed to optogenetics like rhodopsin, but the intracellular part is still kept like the adrenergen receptor, like a beta 2 adrenergen receptor or F1 adrenergic receptor. I mean, like the outside part is the rhodopsin, rhodopsin, and the intracellular part still keep like beta 2 adrenergic receptor, alpha 1 adrenergic receptor, or like the serotonin receptors. And then the intracellular part will keeps those ligand G protein coupled receptors, and after activations, the intracellular part will cross the subcellular signal pathways activations. And the RGS2 is not directly with the G protein, is with the RGS2. It's like accessory proteins that can help the GQ proteins to dissociate to actually it hydrolyze the GTP into GTP and causing the activation of the G protein. So how this function is that, so at the resting state with ligand stimulations, all the system and GQ systems at work. After shedding the light, the CIBN were binding with the Qui2, and this will cause these R2 boxes were very close to GQ, and it causes the inactivation of the GQ and stop and terminate the calcium effector signaling with the light. So this is an opto-RGS2 system. It's like using the light to terminate the signal of the G proteins. Another is the optoprotein or optobody, and this experiment is published quite new in 2019. It's actually a binding with the binding recognition site using an antibody. So with the light, they will cause the antibody to form of functional that's complex, and it will binding to the G protein coupled receptor and cause this receptor to be activated because the intracellular part is being covered by this optobody. So last time in previous lecture, it's mentioned this optobody, but it's not very so clear. So I just ignore on that part. But this chapter it clearly shows that it's based on antibody, and it's using a light to activate these antibody functions and stop the function of the G protein coupled receptor. And this 2014 result is using the light to cause the receptor tyrosine kinase to form a dimer and start protein-protein interactions, and using light to activate this kinase signal. So there are several examples like 2016 Endo lab also using the same way, that's using the light to activate and it causes receptor dimerization, and this activate the growth cone signal pathway and cause the growth cones to grow with the light stimulation directions, means that this part of signals is being activated. And also in 2018, a ligand binding the TRK induced receptor dimerization. It forms a dimerization. So, and how to to improve this dimerization, and it's been discussed by 2013 Duan's research work. Here he discussed about that the extracellular part of the CRY2 actually is determined how the protein interaction between these two, how it works, and how, because some parts of this of the proteins itself, it may inhibit or may reduce or decrease the protein-protein interaction. So for the cluster, the best one is that induce find that's the CRY too high, and find that this compositions is even better than the Y type. And they also find CRY2, they just make many mutations, then to finally find out that CRY low that can reduce like the homodimerization of the protein interactions, and that's so this construct may be helpful for the heterodimerization of the CR2 with the CIB1, and this construct may be important for like the homodimerization between the same CRY2 proteins like this. So as for the TRKA, a previous report suggests that a developed molecule using full-length TRKA cannot be fully activated by CR2 oligomerization, partially due to the inhibitory effect by the extracellular and transmembrane domains. So and here this is another function that's using the light and cause these tools to dissociate with each other. So for the light-induced dissociation module, for example, MXCBD and the TDCBD, it's fusing with the membrane tether intracellular domain of the fibrous growth factor receptor 1. So also, the authors also discuss about the optogenetic system for control receptor traffickings. So this is like a lysosome, endosome, like some small vesicles within the cells. So, and this Rep5 is expressed on these vesicles and the endosomes, and these vesicles were fused with the CIB1. So after the light, then this stimulation and the CIB1 will binding to the CRY2, and then all these endosomes are clustered together. So this segregation will reduce endocytosis rate of EGFR, which will be monitored by dye-labeled EGF. On the other hand, light-induced aggregation Rep7 diminished regulation of EGFR in the latter compartment. So this compartment aggregations were like a pause the cell functions related with the growth factors receptors. So let's just stop there. So it will reduce endocytosis rate of this receptor, and it will also reduce the degraded function of these receptors until you see another signals that to dissociate those endosomes. So that's how those light control can control endocytosis and exocytosis functions. So another is the light-dependent GPCR endocytosis, and this experiment designs is link the CIB1 with the beta-arrestin. So after light stimulations, the CRY2 were formed on heterodimerized with the CIB1. It will cause endocytosis of the receptors. So it is another way to control receptor traffickings. So here and in the authors also introduced an optoligand TCR system. So in this TCR system adds the T cells receptor systems, and from fin B tetramer will generate often with the light, then the fin B will link binding with the PRF6, and it forms the TCR receptors forms like closer together, and were induced to segregate together. So this segregation will trigger the calcium influx and will lead to T cells activation. So this is another related with the T cells receptor functions. That's this is the material like the extracellular matrix, and the T cell receptors were linked with the SLV2 of the light. Will dissociate with the ZTK. That's the LV2 and ZTK system. Light will cause the dissociation, and this dissociation will cause an activation of the TCR receptors. And the blue light dissociation between will reversibly decrease. Screen share stop association cause. Okay, then I will go back. Light dissociations. So also chemokines. So in the figure C, in the talking about a light-dependent social modules, it can also use as to control the ligand release upon the illumination. So within the ERs at the resting state, the interleukin were fused with the UVR8 tandems with transmembrane sequence, and they were forms like binding together. So after shooting light, those dissociation between these two different, with several different UVR8 structures, and can help these help these molecules to traffic into Golgi compartment, and within the Golgi compartment it can be cleaved by furin and it cause a release, and this will be enter into the vesicles and for transporting to ready to be released to the other cells. So you enter the UV radiation, dissociated chemical will derive to OH, or chemical will cleave off by fluorine, and light-induced chemical sections will be confirmed in mouse and zebrafish. So in the beginning of this lectures, we're talking about a protein-protein dissociation system, that's the PhoCl. And here, so the authors discuss about how to study the cell mechanical interaction and signals using the cadherin. As in here, that link the cadherin with the PhoCl using the light, and it will cause a dissociation with the cadherin, the transmembrane part with the intracellular part. So, and the later you were like using a light to stop the cells crosstalk between two different cells. And also another already described system is law of the integrin optometrics. And the optometrics that were fused with the PVB at the cell adhesion surface, and the integrins can be linked with PIF6, and using with the light, and these two binding together, and the light can induce a cell adhesion, trigger downstream mechanical sensory pathway. So start the signal crosstalk between the cells with the extracellular matrix clusters. So that's all. That's so these lectures were actually previously discussed about emphasize on how the light control the protein-protein interaction and protein-protein dissociations. And it's more clarified about the information about the optobody, and also mentioned about how those cells interactions with the actual cell matrix. And I think it's quite interesting. Okay, that's all.